This is a great honor to invite our speaker today, Dr. John Cherry. He's given a talk in exercise physiology for us for the past, I don't know now, what, 3-4 years? And every year it's one of the highlights of the semester. Everybody raves about it and always asks me a lot of questions afterwards. And I'm not qualified to answer those questions, but he is. So feel free to write down your questions and ask at the end if they come up. But so Dr. John Cherry, he is an assistant professor of pathology and laboratory medicine at Boston University's School of Medicine, and as well as he leads the digital pathology hub at the BU CTE Center, where you'll see some of this work. Without any more delay, Dr. John Cherry.
Well, thank you for having me. I'm always happy to give these talks. I think everyone can learn a lot, even myself, answering questions about these. So very happy to come give these talks all the time. This talk is structured for everybody. It's going to give a lot of history information, a lot of recent information. So I think this is something that if you don't know anything about CTE, you're going to learn a lot of sides of it. And if you already know something about CTE, well, I think you can hopefully learn a little bit more from this talk. So I think this is going to be a good talk that covers most of the aspects of it, so hopefully if you have a question coming into this, I can answer it, and if not, please let me know. We can talk about it later. So this is going to be the structure of our talk. We're going to talk about the history of CTE, which I think is a really often overlooked area. Then we'll go into what is CTE. That's always the question. What is it? The clinical identity is what we'll talk about next, which I think a lot of people have a lot of interest in, so we'll really get into the details there. Then we'll go into the science behind it, which is what a lot of my work is focused on, the science behind the disease. We'll go into detecting CTE, which kind of goes hand in hand with the clinical identity. How do you see if someone has it, which is a big question nowadays. And then we'll end with the future directions and hopes. And if you can't hear me in the back, just let me know. I'll try to yell louder. I've never been told I've been quiet, so hopefully I can reach the people in the back. So first, let's talk about the history of CTE. This is not a new disease. Even though we've started talking about in the past 10, 15 years, CTE has been around in different shapes and forms for almost 100 years now, if not longer. This is the first reported study by a pathologist, Dr. Harrison Martland in New Jersey, in the early 1900s. Now, Dr. Martland is a pathologist, and he was involved in looking at individuals, particularly boxers, who were of the slugging type. Now, people that really stood in the pocket and hit each other. He found that these individuals who received a lot of blows in boxing had unsteadiness of gait, mental confusion, slowing of muscular movements, and tremors, and he thought this was caused by multiple concussive hemorrhages. So getting hit in the head a lot causes some sort of hemorrhage, which caused these clinical symptoms. So even 100 years ago, we knew getting hit in the head a lot was bad for you. This is not a new thing that we're discovering now. This has been around for a long time, but primarily the early work is boxing, since that's a sport where people can stay in for a long period of time, 10, 20, 30 years, and they get hit a lot. So that was what the focus was. So this was called punch drunk too, which is still a term that you can hear today. It's still used in circles. Now the name evolved over time. In the 30s, it became known as dementia pugilistica, again a term you can even hear today. And Dr. Millsbraw, who coined this initial term, would say that nearly half of the fighters, again boxers, who stayed in the game long enough, would develop this condition. In the 40s and 50s, the name sort of progressed to progressive traumatic encephalopathy and ultimately chronic traumatic encephalopathy, to give it a more scientific nomenclature to the name, because punch drunk and dementia pugilistica, they don't really sound good in terms of clinical term. So again, this is the 50s. CTE is a thing in the 50s. This is not a new disease. But all of these early studies are clinical based, meaning that someone saw a boxer, in most cases, had a problem, and they said, hey, this person boxed and had a problem, therefore they might be connected. There was no real detailed study on let's look at someone's brain and see if there are any correlations to damage in the brain that we can see. So it's all clinical up to this point. Until about the 70s. We have Dr. Cresselli publishing this case series of 15 retired boxers. This is the first clinical and neuropathologic observation and documentation that there's record of. And these brains still exist today. You can go and look at them now. It's a really, really fascinating case series. But this is the first time you can ever say, hey, there's something going on physically in the brain linked to these people with these clinical symptoms. So now we're actually linking them together. And again, most of this is in boxers. It's not really wide known. It's known to the people who are in the industry, who do some of the research, but it's not really a public interest, a public concern. Because it's boxers. They box too long, and of course they're going to have issues later in life. Until about the early 2000s. Now, this is the case of Mike Webster. He died at the age of 52. He played in the NFL for 17 years, and he played college football and high school football before that. So he has a lot of exposure to football. He was pretty significantly impaired. He had a lot of cognitive loss, behavior problems, and motor symptoms. And when he ultimately passed away, his brain was studied in an autopsy by Dr. Bennett Umalu, who saw that he had what we now know is CTE. And this is really where the story kind of sets into play. This is someone who played, a pastime, a fun sport. They weren't a lifelong boxer. They played a sport that kids play, that kids enjoy, that everyone enjoys watching. Right? This made it a much, much bigger concern that if you play this sport long enough, there's a potential you're going to end up with dementia. End up with this degenerative disease that shouldn't be part of this fun pastime sport. And that really set the field off on how big of a problem this is. So where do we get into this? Where does BU come into the picture here? Dr. McKee, Dr. Ann McKee, who runs the BU CTE Center and the Alzheimer's Disease Center now, she is a neuropathologist up at the Bedford VA Hospital. Dr. McKee is an expert in Alzheimer's disease, and that was some of her primary research. And then a case from Paul Pender, who is a world champion boxer, a local guy, came across her death. He passed away of severe dementia, died at 73, and when Dr. McKee looked at his brain, he had anything but Alzheimer's. He had very striking pathology that she's never seen before, and it ended up being CTE. And that really set her on the path of wanting to know more about it. She found experts in the field to talk to about it, and ended up hooking up with a lot of other experts in the area to form what we now know as the BU CTE Center. And they really set to work in characterizing the disease. Like we talked about previously, a lot of the early studies are just clinical or these small 15 individuals, if not less, case studies trying to show what a disease is like. It's really hard to learn something from very, very small studies. Is it representative? Is it not representative? What are you looking at here? So one of the first major accomplishments they had after looking at CTE came in 2013. When they published the, at the time, largest case series of neuropathologically autopsy cases with CTE in 85 different cases that had CTE. This was the first in-depth look at the neuropathologic features that define the disease. Never has been done before this. It was only those small case series. And even at this point in 2013, CTE is not recognized as a unique disease. There's a lot of naysayers at this point. There are now, but at the time now, or at the 2013, there was a lot. This is a weird presentation of Alzheimer's disease. You're looking at normal aging. You're not looking at something right. This is just misstaining. The works of criticisms about it. And this is after this huge case series. So what do you do to combat this? Well, you bring in experts from all over the world who will believe you, don't believe you, disagree with you, are on your side. You bring them all together and do what was a consensus meeting. You bring in all these experts from across the world into BU, and then you have criteria. You talk about what makes CTE potentially a unique disease. Then you start looking at case series. You start looking at diseases that resemble CTE, that don't resemble CTE, trying to diagnose it, trying to see what makes it different from these other pathologies. And I won't go into this whole study here because it's pretty intensive. But what they ended up showing is, again, with people who believed it and didn't believe it all together, that yes, this was a unique disease. This wasn't atypical Alzheimer's disease. This wasn't normal aging. This wasn't bad protocol, bad staining. This is a unique pathology, and this was the first time in 2015, not even 10 years ago, that it was recognized as a unique pathology, not just a quirk from one little group in Boston. So this is a very significant part of the field. Again, it's only less than 10 years ago, so that's how new we are in the research right now. It's the cutting edge, essentially. And I like to show that based off the amount of publications that there are on CTE. So this is just a PubMed search of the amount of publications that have CTE in the titles. And the group that did this set this up into 3 different stages. The initial stage, when you first start to learn about it, there's a couple a year, not too many, it's still a new thing. The second stage, when Dr. McKee gets involved and really starts to ramp up that production. And the third stage, which is what we're in right now, is this exponential increase in science. So we are on the cutting edge of this. That's why every time I give a talk, I can give you new data. Every year, there's significant advancements in the field that is that new. And it's that exciting. And I hope throughout this talk I can show you what some of that work is and really spread the message of what we think CTE is. So with that, what is CTE? Right? We talk about a lot, chronic traumatic encephalopathy, CTE. What is it? And CTE is defined by the pathology. The pathology makes the disease. Because you can't diagnose it in life yet, so we have to understand what the pathology is. And there's two ways you can look at it. The macroscopic features, so the things we can see without a microscope. If I just look at a brain, what can I see on someone who might have CTE? And there's a couple things that stand out. You have what's called a cavum septum lucidum right here. So you have a membrane that goes right down the center of your brain, essentially. And it's a continuous membrane. It shouldn't be teared or divided. But through some trauma-related mechanism, this septum opens up. So cavum, like a cave, it becomes like a cave. Essentially, that's how you kind of remember that. It's not always involved in head trauma. Sometimes it's congenital. But we see it in a lot of cases with someone who has trauma, this cavum septum lucidum. Now, the other thing you can see is this really gross atrophy overall on the brain. They have a lot of shrinkage on the brain, loss of tissue. You get these big cavitated spaces coming into the brain. And this can be seen on very young individuals, too. So this isn't a brain of like a 90-year-old. You can see things like this happening in people who are 50, which is really, really, depressing in terms of how this affects people. It affects them so young. The other thing is you have small hippocampus. So again, when you're seeing more and more holes compared to a normal brain, the brain should be very flush, pushing out on things, no gaps. When you start seeing these gaps and holes, that's a problem. That means you have tissue loss. But these can be found in other diseases too. These are not specific to CTE. So when you're looking at the brain, there's things that can give you points that this might be CTE, but it can never be definitive. It could be Alzheimer's, sometimes other temporal dementias, but it says there's something going on. So what you have to do is look at the microscopic features of CTE. And the specific pathology that's associated with CTE is tau pathology, hyperphosphorylated tau. Tau is a microtubule associated protein that's in all of our cells. It's essentially a scaffold protein. So it's inside your cells and provides support and stability inside of those cells. So it's really important, we need it. Through some mechanism we're still trying to understand, those individual pieces of tau protein become unbound from the scaffold and they get sticky, they get phosphorylated, what it's called. That phosphorylated tau, or p-tau as we call it, can start binding and clumping together with other p-tau. And that can cause more tau to come off and get sticky, and more pathology, and more tau to come off and get sticky. So you end up having these clusters of tau, which are seen here in these stains, that are indicative of the disease. Now, tau is found in other diseases. There's a whole class of diseases called tauopathies. Alzheimer's is one. There's a lot of them. What makes this unique to CTE? Well, one of the things that came out of the consensus conference, and a lot of the work beforehand, was what's called the pathognomonic lesion. It's a feature that defines the disease, and it's three things. Three things, and it's not very complicated when you break it down. I'll just say it once without breaking it down. It's the perivascular accumulation of hyperphosphorylated tau at the depth of the cortical sulcus. It's a lot of terms there. Breaking it down, it's a lot simpler than that. Perivascular accumulation, it's around blood vessels. That's all that is. Hyperphosphorylated tau, we talked about what it is. At the depth of the cortical sulcus. Your brain has the peaks and valleys in it. You've seen the folds. At the bottom of those folds is your sulcus. So what that is, perivascular accumulation of hyperphosphorylated tau at the depth of the cortical sulcus. Tau is around blood vessels at the bottom of the folds. That is not seen in any other disease. It is the pathognomonic lesion. It defines the disease. If you see it, somebody has CTE. No questions asked, full stop. Now, the other thing that CTE is patchy. Some diseases like Alzheimer's, you get pathology throughout the whole cortical ribbon here. So you'll see a big line, and it'll be everywhere in the brain. CTE is patchy. You'll start to get these patches at the sulcus here, so bottom of the folds, in certain areas. So you can see here, here, here. That's one of the features of it. It's a very patchy disease for whatever reason. Now, based on a lot of the early criteria, they set up 4 different stages for it. So what's low, medium, and high, essentially. And it's based on where the tau is. 1 or 2 small foci of tau in the brain is clumped to stage 1. When you start to get a bit more throughout the brain and just in the cortical areas, it's stage 2. Once you start to get a deeper involvement in your hippocampus or your memory centers, that's stage 3. And then stage 4 is just brain-wide involvement. This was the first way they could classify it. And this was part of the original 2013 criteria. But things need to get updated and improved. And we're always trying to update and improve. And I think you'll see this in the talk, is I'm going to keep saying, and then we did another study. And then we improved it even more. And that's what we did. We did another study, and we improved this criteria even more. So just like we had that original consensus meeting in 2015, another one happened in 2020, 2021. The second consensus meeting. And we don't need to go into all the stuff, but the same people from the first one came back. And they said, okay, let's look at all the research and all the science, and what's changed? What have we found? What have we improved on? And there's several things. One of the most important things they did is they simplified the criteria to diagnose the disease based on the pathognomonic lesion. Instead of having stage 1, 2, 3, 4, and how much tau is here, how much tau is there, where is it? The criteria became a lot simpler. Is there a lesion, yes or no? If there is, how many places in the brain do you see it? How many places do you see tau? If you have 5 or more, you're high stage. If you're less than 5, you have low stage. It makes someone who's not trained in identifying this much more likely to have a consistent diagnosis over time. So we simplified the criteria a bit. The other important thing we did that's not on here is we clarified what the cells are involved. So there's both neuronal and astrocytic tau, so different cells in the brain can have tau pathology. One of the debates in the field is, well, what cells is it in CTE? And something that came out of this study is, well, it's more neurons that have tau pathology. And astrocytes are more of a different disease, it's more age-related. But that's not as important here. It's very important for the field. So that's one of the updates that we had. Now, with that, we can actually start talking about the clinical identity of CTE. And I think this is what people are very interested in. What does CTE look like in life? Well, I can't tell you exactly yet because you can only diagnose CTE after somebody dies. Someone who is alive, can you see that pathognomonic lesion? You can't. How do you diagnose CTE? Because that's the only way right now you can diagnose it, is to see that lesion. So if someone is alive, you can't diagnose CTE yet. However, there's a lot of work trying to understand what CTE in life looks like. This is the first attempt at that in 2014. Again, only 10 years ago. This is a literature review trying to classify what the clinical features of CTE actually are. And the term for CTE in life became traumatic encephalopathy syndrome, or TES. So you can't have CTE while you're alive, but you can have TES. So it's a way to take it apart from the pathology. Because the pathology is a pure pathologic diagnosis, and the clinical diagnosis should be separate from that, because we can't make it definitive yet. So this original study proposed several clinical features that are associated with CTE, or sorry, TES, and you're seeing here there's a lot of them. So behavior, mood, cognitive, motor, and this is what you typically think of when someone says, well, what is CTE in life? Explosivity, loss of control, loss of fuse, impulsivity, physical violence, and that, these are just a lot of symptoms that have been shown to be possible in CTE, maybe not associated with it. So what is the criteria for actually diagnosing, or I should say the original criteria for diagnosing TES? So this is the first pass at it. This is what the first group decided to make. And it comes down into 5 different criteria to get a diagnosis of TES. The first is having a history of multiple impacts. It can't just be one trauma, and that's it for your life. It needs to be a history of multiple impacts from sports, military service, and should be at least more than 4 years or 2 years, depending on the type of injury here. So that's the first thing that has to be shown. You need to have all 5 of these. The second is you can't have anything else account for your symptoms. So it's going to be essentially a disease by exclusion. Can anything else account for the symptoms, such as, a single TBI? Persistent post-concussive syndrome, which is very different, or I shouldn't say very different, but it's different from CTE symptoms. Other diseases, like Alzheimer's, can't account for the symptoms. So you have to make sure that what you're seeing is not Alzheimer's pathology. Now, substance use can be considered, but it gets tricky, because that comes with its own set of problems. If somebody is always high, if they're always drunk, how do you associate those symptoms with the disease, or not with the disease? What is covering up what? So that makes it very tricky. The third is the features must be present, that's very small, they must be present for at least 12 months. So this is a disease that takes a lot of time to occur. This isn't something if you get hit in the head one day and you have a concussion, the next day you have symptoms. That is not CTE. They need to be present for 12 months minimum. Then you have to have a core clinical feature. So that big list of symptoms I showed you, you've got to have some of those features on there. Either cognitive, behavior, or mood. You got to have some of those features that were listed up there. Now, the last thing is supportive features. This is a bit more ambiguous because there's a little bit of wiggle room on what can be called a feature here. And you need to have two of several features. Got to have documented decline, so you need to get worse. This isn't something that stays at baseline the whole time. It has to get worse over time. Then you need to have either delayed onset, or impulsivity, apathy, anxiety, paranoia. So this one gets a little bit tougher. There are a couple things, but you have to have two of these supportive features. And if you have 1, 2, 3, 4, 5, all of these, then you can get a TES diagnosis. And based on what the clinician feels, how you did on those scores, they could diagnose you with either probable, possible, or unlikely. Now, there's one thing we didn't talk about, that's the motor part of this. Motor features they're not a core clinical feature, but they're supportive, and they make things, again, challenging to diagnose CTE, because they could still be present. And the presence of these features can often mistake it for another disease entirely. I bring up the case of Kevin Turner. He was a Pro Bowl NFL player. He passed away several years ago now with ALS, so motor disease. Everyone was sure he had ALS. It was so tragic. When they looked at his brain, he had CTE. He didn't have ALS at all. He had CTE in the area of the brain where ALS can occur. So that complicates diagnosis, doesn't it? Diagnosis gets even more complicated. There are modifiers of symptoms, clinical modifiers. We've shown that the age you first start playing sports can result in worse impairment. There's a thing called cognitive reserve, so the more education you have, the more protected you are from showing cognitive issues. So you have a longer grace period, essentially, before you start having problems. You can have other diseases with CTE at the same time. Often people have multiple pathologies. They might have CTE and Alzheimer's disease, CTE and Lewy body disease. In fact, sometimes when you have one, it makes the other more likely. And they have their own set of clinical symptoms. So how do you diagnose this disease with all of these things occurring at once? Pretty challenging, isn't it? You got to validate this in a large sample set. You need to do a very particular kind of analysis. So using this original criteria, what we started to do was clinical pathologic correlation studies. This was a very intense study led by Dr. Jesse Metz here with collaboration of Dr. Ann McKee, where the idea is to marry the clinical and neuropathologic features of this disease to try to understand what clinical features best correlate with the neuropathology. So we have this huge brain bank with hundreds and hundreds of brains that all have clinical information on them. Can we utilize that in the best way to find the clinical symptoms that are most related to the disease using this TES criteria? And that's what the study does. The way this works, someone passes away and donates their brain, it gets a full neuropathologic workup. So we know what the disease is after they pass away. We then have research associates go back to the family and do a full in-life history. We get in-life history, medical records, sports records, any sort of clinical records we can for this individual. And a sheet is written up about their life. That sheet is then read to a clinician. Clinician's job is to listen, understand, And then they will try to say, well, based on X, Y, and Z, I think this individual has this disease or this disease. We could then say, okay, you are incorrect or correct. And that's one data point. So now we have symptoms X, Y, and Z that he guessed were correct for CTE or not. We have to do that hundreds and hundreds, if not thousands of times. Then we can start to correlate all that data together to see what works best, what doesn't, to diagnose CTE. Let me give you an example of this. What a clinician would receive is a script that would essentially say this is a 25-year-old college football player. They played 16 years of football, 3 years at D1 as linebacker special teams. They had multiple concussions, vision changes, memory problems, confusion, difficulty sleeping, headaches. They quit football after 3 years in college, but they continued to have memory problems, disorientation, attention, concentration, and word-finding issues that lasted for at least 2 years over time, and it got worse and worse over time. They had depression, impulsivity, anger, and they ultimately passed away from a staph infection at the age of 25. Now, using all this information, the clinician would look at this and say, this is a very young individual. They had multiple bouts of head trauma over a long period of time, 16 years. Even after they stopped playing football, they had problems and they got worse over time. Therefore, we think they have CTE. And there's a lot more information in here that we're not discussing, but that's kind of the gist of it. Then the neuropathologist will come and say, you're correct. They have CTE based on this and this. They had stage 2 CTE using the old criteria. Again, this is a single data point. We have to do this thousands and thousands of times to get usable data points in here. And that's what was done for this original TES criteria. And we want to see how well did it work. How well, using our original criteria, could we diagnose CTE? How good is this? And a couple years ago, 3 years ago at this rate, we looked at the validity of that criteria. How well did it work? And what it came out showing that the TES, the original criteria, provided good evidence to rule out, but limited evidence to rule in CTE pathology. So essentially, what we've done is we created a very sensitive test to if somebody has a problem, if they have a degenerative disease, but it wasn't very specific to CTE. What we could show is that, yes, we can most of the time catch, does this person have a neurodegenerative problem of some sort? Whether that be CTE, Alzheimer's, ALS, you name it. It was good at that. But less good on the type of disease. So we have to better it. Again, that's what we're always doing here. We're creating criteria, science, doing the work to examine it, seeing how it is, and improving it. So just last year, 2 years ago, we had a new criteria that was proposed in efforts to better the original criteria. And this slightly tweaked what we talked about before. You still have your 5 fields here. So the first one is you need to have a minimum of 5 years of exposure to contact sports, so minimum of 5 years. You need to have your core clinical features. They must be progressive. So that's, again, that stays the same. The new thing is behavior symptoms like depression, anxiety, apathy, and paranoia, they're not part of the behavior phenotype. They're excluded from the diagnosis here. We'll talk about that in a little bit. And again, you can't be fully accounted for by other disorders, so that's the same. And then if these are met, you have TES. Then there's a whole other cascade of what kind of TES you have. So if you are found to have TES, you have to go through this suggestive, possible, probable, or definitive criteria here. And don't get too caught up in all the little boxes there. But the important thing here now is if you do have TES and you don't have cognitive symptoms, you are immediately lumped into suggestive. Because it seems the cognitive component is one of the most descriptive elements of it. And behavior phenotypes end up being very permissive and not as specific to diseases, because a lot of things can have behavior phenotypes. You know, bipolar disorder, many of these other depressive phenotypes, they can mimic these behavior phenotypes, and it's hard to untangle them from the disease. So what this criteria does now is it says we need to have memory or cognitive problems to progress here. You can still get a TES staging, but it's suggestive. It's not going to be the possible or probable, which are based on having more of these other features in there. And again, the only way to get definitive TES with CTE is neuropathology autopsy. So that's where it stands right now. And again, we're doing what we did before with the consensus conference, doing this on hundreds and thousands of cases and seeing how well this new criteria does. Is it better? Potentially it's better. Is it best? We'll have to see. We'll have to see how it looks another 5 years from now when we do the same study. And if it needs improvement, we'll do it again. That's the iteration of science that we're always trying to improve this. Okay, let's talk a little bit about the science behind it now. This is an area that's where my work is very situated in. Really asking the question, how does head trauma lead to CTE? How does this lead to this? What is this middle step? That's such an important question. What is this middle step? And a lot of times the thought is, well, this is just a hit to the head, right? Your head's getting knocked, your brain's getting bruised, and that's the problem. It's this physical blow to the head that's doing it. That's the problem in CTE. And that's not the case. If you get hit hard enough in the head that you have like a bruise in your brain, you're bleeding your brain. That's really bad, right? That is not what's happening here. It's not this physical smack of your head going through your skull and hurting your brain. That is a more of moderate TBI, like or severe TBI even, and you should go to the hospital immediately. So that's the problem. What's happening in CTE, in the damage that will result in CTE, is these forces are acting on the brain. Acceleration, deacceleration forces, linear rotational. So someone is getting hit. They are moving very quickly and stopping, and maybe moving back quickly the other way. The brain is not just moving with the body perfectly. It's moving one way, stopping, twisting, and turning as you're stopping in the order of microseconds and coming back together. Brain's not supposed to work like that. And you have a lot of long fibers that process in your brain that are very susceptible to this kind of injury, this twisting, turning, contorting injury. There's a thing called, you have axons in your brain, which is long structures that run from, potentially all through your brain in some areas. When you start turning and twisting them, they get damaged, and you get what's called diffuse axonal injury. And that, you can either cut the axon or cause a neuron to die, and it can lead to long-term problems. There's a lot of things that are occurring in the brain. This is one of the things that could be occurring here. Now, that leads to the question, what about helmets, right? Do helmets actually do much? If we're talking about this sort of biomechanical injury that you're getting after trauma, the physics of the trauma. Let's think about it. You have a brain. Put the helmet on a brain, and you get hit. What is again happening to the brain? It's moving in space and time. It's moving forward and back, right and left, side to side. What is the helmet doing to stop that? Not a lot. Sure, you can have some neck support. Sure, it's stopping some of the actual physical impact. But this biomechanical reaction in the brain is not being accounted for by the helmet. The helmet protects your skull. It is not doing much to protect your brain. So whenever we have these talks, it's always, well, what about the helmets? They're not doing a lot, at least for this particular type of injury. It's not saying don't wear a helmet, because it does protect you from breaking your skull. But in terms of stopping this biophysic injury, it's not helping you too much. Now, what's really interesting about this sort of linear rotational injury is that there are certain areas of your brain that are more vulnerable than others. They're more vulnerable to this mechanical stress. And there's 3 areas that are doing this. The depth of the cortical sulcus, the white matter-gray matter junction, and blood vessels. 2 or 3 of those should sound familiar. It's because they're part of the pathognomonic lesion. So right there, we linked head trauma to CTE. Where CTE is the strongest linked area to injury. So you just linked it right there. Now, why is that the case, though? Well, this is a great example. Dr. Lee Goldstein showed me one time. You take a candy bar, those little ridges, almost Halloween. I think we've all seen candy then. When you rip it, where does it rip? Right down at the bottom of these ridges. Those ridges are there on purpose to concentrate force into one area to make it easier to rip something. Your brain has these peaks and valleys in it. Same thing. when your brain is moving and twisting, where is the stress most severe? The bottom of the sulcus, right at the sulcus. What about blood vessels? You ever look at a manhole cover? Directly around it, you're always going to have cracks. That's because you have two different types of tissue or two different types of material directly abutting each other. And between them is an area of vulnerability. It's an area that we're forced to get concentrated and cause the biggest injury or biggest crack in the case of a manhole cover. Your blood vessels. You have brain parenchyma and your blood vessel. Two different types of tissue directly abutting each other. When you have force directly applied to that, right at that interface is going to have much more severe force concentration and injury is going to be there. So that's why depth of the cortical sulcus, perivascular accumulation. Those areas are more mechanistically vulnerable to damage. Now, this isn't a doom and gloom talk. One injury is not sufficient to cause this. Right? The frequency of trauma is really important there. Our brains are very good at repairing damage. But we're talking about sports that are very frequent in their head trauma. Now, people can report concussions in all these sports: football, soccer, wrestling, boxing, rugby. And there's a very important thing now I need to talk about, is we just talked about concussions and head trauma, but the most important thing I've tried to get out of this talk is that concussions are not related to CTE. We're talking about injury to the brain. A concussion is a clinical event. You got hit in the head hard enough to have a response. We talk about all these sports: football, soccer, boxing. People aren't getting concussions every game. They're getting hit in the head every game. They're getting hit in the head every practice. They're getting hit in the head all the time. It's those subconcussive hits that are the true concern. So again, I'm going to say concussions are not related to CTE. There's a movie called Concussion about CTE. Concussions are not related to CTE. So it's a very important point I try to get out there. And like we said, it's a clinical symptom. A concussion for me is very different than a concussion for all of you. Okay? So that's a very big criteria here. So that's why it's a very hard measure. They're much more frequent. And we have cases in the brain bank that have never had a single documented concussion. Just they've had a history of head trauma. So it's not a concussion. It's the subconcussive hits that are most important to CTE. The hits that you get hundreds, thousands of over the course of your career, that's the problem. And we did a lot of work showing that this is the case. We can even show that the more you get these subconcussive hits, the more likely you are to get CTE. This is a study by, again, Dr. Jesse Metz, where they wanted to model the likelihood of someone getting CTE based on how many years they played sports. So not number of concussions they had, how many years they played a sport. That's the only metric here. Did they play 1 year versus 10 years, essentially? He looked at 256 participants, 223 with CTE and 43 without CTE. And they examined years of playing and the presence and severity of the disease. And what they saw is that each year playing football, they had a 30% chance increased risk of developing the disease. That risk doubles every 2.5 years, and the risk of severe disease doubles every 5.5 years. And that's additional years of playing. We could show that when you play less than 4.5 years, you had a 10-fold less likelihood to get CTE. Played 14.5, you have a 10-fold more risk at getting CTE. So this is just using the number of years that you play a sport. That's it. That's the only metric here. Now, this is fairly scary numbers, but there's something to consider here. We're talking about risk factors, not gotta-have-it factors. Right? There are many, many other factors that play. Just like smoking is a factor for lung disease, doesn't mean someone gets lung disease. They're more likely to get a lung disease if they smoke. But there's a lot of other things here. We had 9 cases that played less than 4.5 years. They still had CTE. And 2 cases that played for more than 14.5 that never got CTE. So there's genetics, other environmental factors. A lot of stuff is at play here. So it's not just you at 14 and a half years, you're going to get it. That's not how it works. Right? We'll talk a bit about genetics at the end of this too. Now, again, just like we keep saying, we tried to make this a better analysis. How do we make this better? How do we improve on this metric of examining who's more likely to get CTE? And we increase the sensitivity of this repetitive head impact, or RHI, measure. RHI lacks some things, obviously. It doesn't account for the type of sport, it doesn't account for the position played, and it doesn't account for the number of actual hits. So how would we go about doing this? Well, we can leverage accelerometer data. There's devices you can put in a helmet and measure this person got X hits at this force. We can make that position specific. This person played lineman, this person played quarterback, this person played kicker, this is a wide receiver. We can add all that information to this measure to make it more sensitive and predict who is likely to get CTE based on all of these factors. And we just recently came out with a paper that is doing just this, making this measure more sensitive. And we implemented what was called a cumulative head impact index, or a chi-score. This chi-score takes into account the career length, so how long you played, it's still an important number here, the position and the level of play. And it also incorporates what's called a positional exposure matrix. So this characterizes the exposure better. It takes helmet sensor data and adds that into the model for specific positions of play. This is just for football now, since that's a lot more published and well understood in terms of positions and accelerometer data. So we take that pen, add the chi, and we have a much better measure of trauma. And then we can look at the biomechanics again. We can look at linear and rotational injury now. And what do we see here? This might not be a very impressive graph to you, but what this shows is that when you look at just the red line here, this is just years of exposure alone. When you add in total hits and G-force, so this chi-score and the positional exposure matrix score, you get a much more or significantly better measure of trauma. You're better capturing people who get CTE and the severity of it. So we're improving the score now. We're improving our ability to both be specific and sensitive to identify somebody who might have CTE based on just their sports history. And we're still working to improve this, because it can always get better and better and better as we get more data on this. So let's talk about the mechanism of CTE now. We've talked a lot about this leads to this, right? I think we can pretty definitively say, that head trauma leads to CTE. But what is the middle step, right? What is causing this? And that's what my research is focused on. And one of the things that I think is driving CTE is neuroinflammation. Now, this is neuroinflammation or brain inflammation. Same thing. It's a fundamental response. It gets a bad rap. Neuroinflammation gets a very bad rap that everyone always thinks that, well, you have inflammation, therefore it's bad. Right? Inflammation is always bad. Get rid of it, take an Advil, put some ice on it. It's always bad, get rid of it. It's a fundamental immune response. You need neuroinflammation. You need inflammation to deal with infections, promote recovery, resolve tissue damage, and normally it goes away. Once the job is done, it goes away. The body has great mechanisms to limit it and make a more resolving response and fix damage. Chronic inflammation, inflammation that's not resolved, that's problematic. And that's what we're talking about with CTE here. Now, when we talk about inflammation, the main cell type in the brain that's associated with inflammation are, I shouldn't say the main cell type, one of the most important cell types are microglia. They are the brain's resident immune cell. Just like your body has immune cells, your brain has its own specific immune system. We found in several studies that they are responsible for the neuroinflammation that we see in the brain. And microglia, you can look at them in a lot of different ways. Normally when they're at what's called a resting state, so not active, they're these very small cells, they have these very fine processes, but when they become inflamed or activated chronically, they have these very much larger cell processes, the bodies get bigger, the processes get shorter, And you can very clearly see that. We see that in individuals that have CTE compared to not. We even see it in individuals with just exposure to trauma, so they just have head trauma, but not yet CTE. So you're having inflammation occurring before pathology occurs. Before tau is there, inflammation is there. And you can see this in other cases too. So there's other diseases that can mimic this. And based on other diseases, what we think the mechanism of CTE is essentially a positive feedback loop. You have an initial event that causes neuroinflammation. Inflammation then can cause tau to be hyperphosphorylated. Remember, tau comes off the tubules, gets sticky, clumps together. That tau is inflammatory. It can cause more inflammation. So now you have a feedback loop starting where inflammation causes tau. Tau causes inflammation, causes more tau. And it can occur over and over and over. And if your body can't fix that, you're going to get a degenerative disease, a degenerative tauopathy. And that's one of the ways we think CTE occurs. This is not everything. This is a very simple diagram of what we're thinking. There, you know, if I wanted to be complex about this, it would have 300 lines of different other things here that are also occurring. But generally, this is what we believe is the main architecture here. And that's just stat line. We can see inflammation in living people. So there are ways to look at inflammation in the brain using PET scans. And you can inject these radiotracer dyes into individuals that will bind particular targets in their brain. And then when you look with a PET scanner, you can see if they have more of this dye in their brain, they have more inflammation. This is work done by my colleague, Dr. Julie Coughlin, where she did this in individuals who were healthy or they played football, age match, so same ages. And they could show that individuals who played football had more dye coming into their brain, meaning they have more inflammation. Now, I don't know if this individual had CTE, because we can't see a lesion here. All we can say is this person played sports, he played football, he has more brain inflammation compared to somebody who didn't. And I think that's pretty significant. Now, I keep saying inflammation. Inflammation is a huge term. It's a giant term. It encompasses so many things. I pulled this from a company website that looks at a couple small parts of inflammation. It is such a complicated field that there are courses and courses and courses dedicated to just neuroinflammation. I've studied neuroinflammation for 10 years, and I'm still trying to get a handle on it. It's very complicated. So when I say neuroinflammation occurs, that's me hand-waving. That's saying, well, something's occurring that's inflammation-based, because it's not super easy to tell you all of the things that are occurring here, but we're getting at that now. A lot of the stains that we do can look at 1 or 2 targets. We can't look at 1,000 things at once doing regular staining. So we have to change our techniques. How do we characterize inflammation? How do we best look at what kind of inflammation is occurring? We need better methods, and those methods are going to be genomic techniques. Now, just very briefly, what we're going to be doing is looking essentially at RNA, right? So you have RNA and DNA. DNA is the building blocks, the code for all the things that you make. RNA, in this case, messenger RNA, is your, it's a message. The cell says, I want to make X. Go tell the rest of the cell that. It's going to produce this mRNA that's going to go into the cytoplasm, and it's going to say, hey, make this protein. We can look at the amount of messenger RNA there is and have that as a proxy of what that cell is trying to do. So that's all RNA sequencing is, essentially, is looking at the message to make a particular thing. If there's more of it, we think there's more of that thing being made. If it's not there, that thing probably isn't being made. In doing this, we can look up to 20,000 targets at one time. So this is a much stronger way to look at these complicated fields, these complicated pathways, is with these really high throughput genomic techniques. And that's what we did. And you can refine it even more. So there's bulk versus single cell, essentially. Bulk is if I take a whole piece of tissue, grind it up, grind everything together, and look at all the RNA in that one chunk of tissue. Tissue has dozens of different cells there, so what we're seeing is the RNA, the message, from a lot of cells pulled together. However, in the last 5 years, there's new technology called single cell sequencing. which lets us look at individual single cells and the RNA component, the messenger RNA component of those individual cells. So I can say, this one microglia, he makes X, Y, and Z. This other microglia makes A, B, C. They are different. And that's really transformed the field, and we'll talk a little bit about that in a bit. So we want to use this to investigate CTE, this technology to investigate it. And we want to focus on individuals who are very young, because that's a very important part of this work, is looking at someone who is very young and might not have a lot of pathology yet. Can we see what's going on in the earliest stages of disease? We just put a paper out looking at young individuals under the age of 30. And I think the numbers were something like 40% of the cases that we saw that were under the age of 30 had some sort of CTE and CTE symptoms. Now that's not saying everybody under 30 who plays sports is going to get CTE, because it's a very biased sample set. You know, people have problems, they donate, obviously, and people that don't have problems don't donate a lot of times. But showing that 40%, if not, give or take, have CTE under the age of 30 is very concerning. And how do we best investigate that? Well, we know inflammation is occurring in these cases, and we know inflammation and pathology can be very subtle. So we want to use our techniques to look at this, the single-cell sequencing techniques to look at this, and that's what we're doing. We're taking gray matter from the frontal cortex. This is the area of the brain where pathology first occurs. We're going to process it for single-cell sequencing. We're going to look at 3 different groups. Control individuals, they don't have any disease, they don't have any exposure to sports. So they are control in all 2 areas there. Individuals with RHI, so repetitive head impacts. They played sports, don't have disease. Then we have individuals with CTE. They played sports and have very early stage mild disease. We did single cell sequencing on these individuals. And this is what you can see when you do single cell. Each one of these dots here, these teeny dots that make up these big clusters, is an individual single cell. And we can look at the RNA component of that cell. Then by knowing what markers make up the cell types of the brain, we can classify them and cluster them together. So we know each of these colors is a cell type. You have your neurons, your microglia, your astrocytes, and we can see what makes them unique. Now from this, we focus on the microglia. So what we can do is take this microglia population here and further subdivide it to look at them specifically and see what makes them different, essentially. And we can find multiple different types of microglia now. So each of these populations is a genetically, or at least RNA, different type of cell, type of microglia. And when you break them out into control, RHI, CTE, so our 3 groups, you can end up seeing that, hey, there's a population up here that's not there in control. And you see it with RHI and CTE. These are inflamed microglia. These are trauma-associated microglia. Now we have the ability to see what these cell types are. And when we break them out into what genes that make them up, we can see that, this is just showing that there are different cell types. So we see that they're what we're calling these RHI-associated microglia, so RIMs we'll call them. And there's 4 different types of them, 1 through 4. And what's most associated with our CT phenotype are our RHIMs 3 and 4. And when we do some techniques to look at what these genes are doing, so you can do techniques to pretty much condense data down. Instead of looking at 20,000 terms, we can group things together based on common pathways that they express. We can see that these cells are involved in inflammation, regulation, proliferation. So both of these populations of cells are involved in neuroinflammation. Now, why is this significant? These are young individuals. This study was aimed at everybody under 50 in our bank. A lot of these individuals have very, very minimal pathology. And a lot of them, a third of them have no pathology. There is an inflammatory phenotype, a degenerative inflammatory phenotype in individuals prior to developing pathology. So that sort of feeds back into our original understanding of it, that inflammation comes first, causes tau, causes more inflammation. And now we're trying to understand what is that inflammation. And this data is probably a month old. So we're trying to understand that more on what these cells are. And hopefully in the next couple of months, we can get this together. And if you ever want to read it, hopefully we get a paper out next year, you can understand what these cell types are. So let's end with talking a little bit about detecting CTE. How do we detect it in life? it's pretty hard. It ends up being pretty hard to detect CTE in life because you can't physically look at the brain while someone's alive, obviously. And we don't know exactly what we're looking for yet. The field is still very divided on what exactly we're looking for. And we'll talk about why that is in a little bit. So there are a couple ways you can actually do live imaging. So we can't physically look at the brain, but we can do imaging to look at scans, essentially. There are PET scans, which we talked about with inflammation, to look at proteins. So you can do uptake of dyes, and whatever, if that dye binds more to proteins, there's more there. MRI to look at tissue loss, and DTI to look at white matter, which we didn't really talk about too much, but it's a really interesting topic if we ever want to discuss that. It's very complicated to link living imaging, live imaging, with the neuropathology. So one of the things we sought to do was see how well some of the imaging techniques actually correlate with the neuropathology. This is a study I did with Dr. Michaelosco at the CTE Center, where they had 6 individuals who they've done life scans on. So these individuals were alive, and they did scans on them. And then these individuals passed away, and they came to our brain bank, and we did neuropathology on them. So together we worked to see, is what they see on the scans the same as what we see in the neuropathology? Do they line up? So they had their live imaging PET scans, and we had our post-mortem staining, looking for tau. Can you correlate that? When there's more PET imaging for tau, is there more protein staining in the tissue for tau? And the answer is sometimes, but not all the time. Sometimes it looks really good depending on the region that you have. Sometimes it correlates really well, but other areas not so much. So the jury is still out on that. But why is this the case, right? We're looking at a protein, we're doing staining. Why can we not get these to line up? Well, let's think about this lesion. It's small. It's very small. The resolution of these scanners is not there yet to look at this small structure in the brain. If you have one small foci of tau in your brain, it's very hard for these scans to pick it up. The technology is not quite there yet, so it's very hard to see that. Now, the other thing is, what kind of tau are we looking at? I didn't say that before. There's multiple kinds of tau. And that gets really complicated. What kind of tau is in these brains? Well, without getting into too much, tau can exist in a couple different forms based on splicing variants. And we've seen that there's an evolution of tau across disease. It starts as one kind, and then can become another kind of tau towards the end. And it's just based on the amount of repeats are on the tau sequence, so it's either 3 or there's 4. Those are the 2 main classes. A lot of these PET markers only look at one kind of tau, and they don't get a big swath of them. So you can maybe only look at one kind of the disease. So if you have a marker that looks at just late stage 4R tau, maybe you're only getting cases that are here, and you're missing all of these other types. So the field is ongoing. We're trying to design better tracers. So besides imaging, what else can we do to identify it in life? Let's look at biomarkers. So fluid biomarkers, using things that might directly contact the brain, like the CSF, or things like blood and plasma, which are secondary feeders to that. And we've looked for tau in the brain, in the blood before. And this is showing that tau is elevated in the blood when someone plays the NFL. So that's one way. We don't know if this person has CTE. All we can say is they played in the NFL and they have more tau in the blood. So you got to be very at face value with these results, because it's very easy for someone to say, hey, I have a test for CTE. You have more tau, therefore you have CTE. It doesn't work like that. So whenever you're looking at these studies that say they can diagnose CTE, read into what they're actually doing. But we're getting closer, and they're all data points. We got to put them all together at the end, then we can get diagnoses in there. We have other biomarkers called NFL. It was named before the NFL. It's neurofilament light, but it just is very coincidental. It's elevated after trauma. What about inflammation? Inflammation's great, it's so complicated. Let's use it, find a signature. Inflammation's so different among different cases. Well, it is, and we are. We're using it to try to find new biomarkers that are inflammatory related. And I'll sort of speed at the end here since we're running a little short, that we can look at high-plex targets. So this is a study where we use 71 different proteins in tissue to see, is there inflammatory related differences there that we can use for biomarkers, and we found that there are. And we can see that some of them are good to predict CTE. And we use them to look in CSF, and we can say that this particular biomarker, CCL21, is high in CTE but not in AD. So it's helping us specify the disease. And we can do other things like CCL11, which is another inflammatory biomarker. It's high in CTE but not AD. So we're using inflammation to find biomarkers too because it's so complex. And the last thing I'll talk about briefly is genetics. Just because somebody plays sports doesn't mean they're going to get CTE, right? It's a risk factor. All these other diseases like Alzheimer's, FTD, they all have risk factors that make you more likely to do it, and genetics are a big part of that. What about CTE? Are there genetics involved in CTE? Of course there are. Individuals with similar levels of exposure might get CTE at different rates. We've seen that. You can plant a line next to each other. One person gets it, the other person doesn't. Why is that? Genetics. There has to be. There 100% has to be. We just haven't discovered all of them yet. And we have done some very base studies on genetic markers like TMM106B and ApoE, and we see that there are differences. Having the more risk allele of these genes makes you more likely to have severe disease. We haven't found a gene that's more yes or no for CTE yet. Mostly everything we find is severity related. So they're not stopping you from getting it, but they're altering how bad the disease is. So that's ongoing work right now. So with that, let's talk a little bit about the future directions and where this work is going. So the biggest thing we have to do is detect it in living people, right? We have to detect it in someone who is alive. Because how do you treat a disease if you don't know somebody has? How do you treat a disease if you can't see them get better? If you can't say, hey, you don't have CTE anymore? That's such a big hurdle in unlocking therapies, is we have to know if somebody has it or not to start treating it as best we can. We have to dig more into the mechanisms, obviously, so that's where my work is ongoing, is what are the mechanisms, what are the microglia, what are the neuroinflammatory phenotypes that are happening here? The other big question is, once it starts, can we stop it? Let's say we can identify it. Can we stop it? That's a big question. After tau is there and a neuron dies, can you replace it? Can you bring cognition back? We don't know yet. So that's getting the idea of stem cell therapy, replacing damaged and lost tissue. And that's on the new frontier now too. We can stop it. Can we repair the damage? We think so. We have to try it. And a big question, what about gender differences? Something we didn't talk about is the male versus female response. Now, being a brain bank that catered mostly towards football because of our inception, we didn't have very many female brains. And that's very significant because there is a big, big difference between male and female response to trauma, and something we're actively trying to study. We just had our first case of a female who was diagnosed with CTE recently. So it's that, not that they don't get it, it's just we don't know the incidence of them because we're only as good as what the data is. And if we don't have the samples to look at, we can't say anything on it. So a big outreach now is the gender component of disease. Then there's another ethnicity component to it as well. So race might actually make a big deal with this too. There's a lot of risk factors that are different between the races. We don't know what they are yet. Other diseases have shown them to be there, so we know that other diseases show us things that could be likely in CTE, but we are only as good as the data that we have. So that's why the work is ongoing, to expand the brain bank, to get more donors, look at every case we have to fully understand the disease. And that's where I think this work is ultimately progressing. Every year, we're getting more and more on this. And hopefully next time we give this talk, I'll have better answers for you. And with that, but I think we're doing a really good job, is what I should say now. But the first people I have to thank for this work is our donors. With them, this work is impossible. We're incredibly grateful for what I do. So whatever we do, we have to thank the donors. And this work is not just mine. This work is a team of people across multiple centers and across continents. It's a huge undertaking, and again, we're the bleeding knife of it. So it's a lot of hands come together to make this work as it is. And I'm just a single person trying to regurgitate a lot of other people's really, really detailed work. And so they're the people that we need to thank for this as well. And if you ever want to get involved, here are our links. You have my email address. Feel free to just email a question whenever. I'm very happy to talk about it. So with that, I can take any sort of questions.